So welcome to my talk. Thank you everyone for actually coming. I really appreciate it. Now, while you were all having coffee, having a croissant, living your best lives, I did a little bit of a social experiment on my side. Now, you're probably wondering why I have all of this cutlery. And possibly in the next 10 years, I could have broken the GDPR law. Now, that sounds really crazy, but I essentially have 4 people's DNA on this seat. And the idea of it is maybe in the next 10 to 15 years, this could be someone's key. This could be a password to someone's entire life. It's a scary thought, but this is where we're heading. And the idea is this where we want to head in the future? So I'd like to thank the 4 people for their DNA. I'll be taking back with me to South Africa. Thank you. No, I'm joking, I won't. So let's look at DNA cryptography and what I'll be talking about. So the talk will focus on 2 main areas, which is using DNA cryptography for the purposes of data storage. So if you're working in a data center or you're really worried about the environment, this is where that comes in. And the next part is basically trying to understand what DNA cryptography actually is. What is this really cool word that everyone keeps talking about in the cryptography industry? And then going into the idea of using DNA cryptography for our own personal use. So the idea that maybe one day I'll have my own DNA key that I'll be able to encrypt and use for any and everything, right? So let's have a look firstly at why I even decided to do this talk. The first part is because I hate myself. And the second part is because I realized one thing, which is that computational power will evolve, right? The computer we have today will not be the computer we have 10 years from now. Meaning the algorithms we have now are not going to be suitable in the next 10 or so years. As it is, we used to have MD5, and we thought MD5 was amazing. And now it's not. Sorry. So now, unfortunately, we have to rely on the math that we currently have in place. And unfortunately, as Roel mentioned, we have what's called quantum computing. The idea that at some point, computers are going to get to the point where we are not going to be able to beat them. And so we've come up with a few ways of possibly beating quantum computing. The first is elliptic cryptography, which is the art of cryptography by basically division. And the other is quantum cryptography, which is messy, and then DNA cryptography, which is why I'm here. Hi. So, elliptic cryptography and quantum cryptography have issues. They're unfortunately prone to person-in-the-middle attacks, and we've also seen cases where they're actually prone to DOS attacks. So it wouldn't be an ideal. Obviously, we can use them, but there will always be that fear there. There will always be that blurb at the bottom questioning whether or not we should have done this in the first place. So, cryptography, what actually is it? So, just in basic terms for you, there is a huge difference between encryption, which is secure, and encoding, which is not. So, think of base 64 versus AES 256. Base 64 is not secure. You can go onto Google now, and you can get your base 64 in clear text. It'll be absolutely beautiful. AES is a little bit more complicated. It involves a specific algorithm. Now, cryptography is also a bit like computers based on our assumption of the computational hardness of how something actually is. And we also have to remember that cryptography isn't a new thing. It's been around for ages. The earliest existence of it was during the Greeks. So say, for instance, Nea over here was attacking me, and I needed to go to my comrade over here. Then I would have a piece of parchment with a specific substitution cipher or a Caesar cipher, and I would give it to my comrade over here, and my comrade would then be able to relay that information. And this is how the Greeks did it. It's very cool, but obviously it wouldn't work today. But just to give you an idea, cryptography has been in research for a really long time. So, DNA. How do we actually understand what DNA is? So DNA is basically a long chain of molecules, and they're what's called base pairs and molecule types. So if you look at your DNA right now, it's all composed of adenine, thymine, guanine, and cysteine, or ATGC. Now, the way I like to think of it is that a base pair needs to sync up. So G will never be equal to T, but A will always equal to T, and G will always equal to C. So the rhyme, ATGC. Now, what's really cool about this is that we've figured out that if we use DNA cryptography, what we could actually do is we could take your typical DNA sequence and we could then convert it into binary. So adenine would be equal to a binary of 00, thymidine would be equal to 001, and so forth. Now, if you want to think of DNA and the structure of it, think of it a bit like a zip on your jumper. All of these things are meshing together. And that's where the AT and GC comes in, is that mesh or that string that links up. Now, the really, really cool thing about DNA, we're all composed of it, but what's even better about it is that we can store a shitload of stuff in it. So DNA is basically an information carrier. It has a large storage space because it basically holds all of the information about us in it. So information about our race, information about our ancestors over 1 million years ago. It's all in that little DNA string. Now, what we realized is that memory cards and chips, they're simply not going to be sustainable for the next 5 years. So what if we could create a form of storage that wouldn't ever need to be updated? And if we think of these large DNA centers, like if you go to Iceland, and if you're lucky enough to see those data centers there, they take up a lot of space. They also take up a lot of power. And in terms of the environment, they take a lot of electricity and consumption and overhead to actually even deal with those. So what if we could make this more environmentally friendly? And what if we could cut down on our storage? This is where DNA cryptography comes in. Because what we realized is that in a single molecule of DNA, we could roughly store over 108 terabytes of data in it, which is super cool. So what we would do is we'd create a synthetic DNA string, and from there we would then store data inside of it through the ATGC. And then from there we would convert it. So we would first have it as a byte of 01, 02, etc. And then from there we would convert it into the ATGC that I showed you before. Now, it's really great, it's really cool, but the only problem is that it takes a really long time to do this kind of thing. So the reason why, for instance, we haven't looked at DNA cryptography in focus is because of the length of it. So if you look at your cryptography methods like DARES, triple DARES, blowfish, it doesn't take that long to do the encryption and the decryption, but if we were to do DNA cryptography, it takes a hell of a lot of time. And the reason it takes so long is because we're thinking of it as bytes versus strings. So when you're doing encryption and decryption, we do it in terms of bytes. But when we're doing DNA cryptography, we're doing it in terms of DNA strings. And that's where it gets a little bit more complicated and where it takes a hell of a lot more time. And this is where it can be a possible pitfall. Because unfortunately, we're human beings. We're not particularly patient people. I don't know if you've ever tried to drive in the traffic. You would know this. So, how does DNA cryptography actually work? And how have the early researchers done it? So the majority of the research is very new. There isn't a lot on it. The earliest paper I've seen comes from 2017. So it's still a very new realm of research. And there's still a lot of questions around it. So unfortunately, in terms of research, we've kind of got to use what we got. So the University of India and the University of Mumbai were the first guys to kind of look at this and say, how can we do this? Now, the way they did it was through what's called the Java implementation with an SDK. So what they did, now this is not obviously a secure way of doing it because it makes use of a substitution cipher, where you would have your ATGC, which you would then use as your substitution. So say, for instance, I have a PDF, or in the explanation over here, you have a plain text message saying test, then the raw DNA message would be something like TCACGC, and that would be the DNA. So that was how we did the substitution cipher with the Java implementation. And what we used was a BioJava implementation with their APIs. And the way we did it was through a getBytes method, which then would return an 8-bit ASCII string for every character that we wanted to then encrypt. Very cool, but unfortunately, this implementation works, but it's not necessarily secure, because obviously, if you know what the substitution alphabet is, you can then do that decryption. So it's not ideal, but it's cool. Now, let's think about DNA symmetric cryptography. And they also did this through the Java implementation, because what's really cool is that the BioJava architecture already exists. It's used a lot in the biotechnological field when they're doing all sorts of like research, app development, anything. They'll use the BioJava framework along with their symbolic alphabetic API. So what they've done here is they'll use a 3-step process to do it, where they'll create the keygen, then they'll encrypt, and then they'll decrypt. And they'll usually do this with either an OTP or a substitution cipher like we've seen before. Now, this is very early in the research stage. It took a very long time to do. And unfortunately, when we did this, we had to have access to public libraries to do it. Meaning, for instance, if you are involved in any kind of medical, So say, for instance, you got cancer and you wanted to be a part of a medical trial. You would then offer up information about yourself, and that information could get stored in a public DNA library. Now, what we do as researchers or those in the biotechnological field is then we would, at some points, request access to those public DNA libraries. In South Africa, for instance, because of the Cradle of Humankind, we can actually access public DNA records of that nature, which we then used to do the research. It was very hard to get access to that. There was a lot of planning. And because of POPI and GDPR regulations, it was very difficult to get access to those libraries, which is also what makes this kind of research particularly difficult to do. Because unfortunately, no one wants to offer up their DNA willingly. As you can see by the cups and the plates I have over here, I'm sure those 4 people aren't exactly impressed with what I've done. And I don't think you'd be particularly impressed if I took all of your DNA, in theory, to do my research. So the way in which we did the DNA cryptography using the OTP is that one key was used for encryption and decryption, and we tested it with DES, RC2, and 3DES, where we took plain text data, we then combined it with a public DNA library, and then from there we added what's called a random key or pad through a SAW operation. It was very cool and it worked. It just took a hell of a lot of time, and many computers and hearts were broken in the process. Now, we then have a more secure version, because what researchers wanted to do is they wanted to remove interaction between the user and key authorities. Because what often happens with asymmetric cryptography is we have the impersonation of key authorities, and this is where attacks happen. So we looked at asymmetric cryptography using RSA, and what we found was that it worked, but it was very computationally intensive. It took us a long time to actually do this. And so we thought that it wouldn't be an option for us in the long term, just because of the speed at which it took. Now, what are the pitfalls for DNA storage and DNA cryptography as a whole? Well, there's a couple. So DNA storage, firstly, is incredibly expensive. It's an incredibly expensive form of research to do. It costs anywhere in the millions to do in South Africa. And what's worse is that there is a high risk of DNA contamination. So earlier I was speaking to a colleague and they asked me, well, Tayla, if we use a synthetic DNA string and we put it in a data center, what would happen? I was like, well, it would be stored. He was like, okay, great. But what if Amazon had data in a data center and Google had data in a data center? How would we ensure that contamination doesn't happen? Well, we don't have an answer for that. It's a huge issue. Like, DNA contamination will be a huge issue. The other thing is that there would be possible data leakage. And so you would have GDPR on steroids. It would be great. And the other issue is that there's lacking research around it. So I'm one of maybe a few people that are looking at this research, unfortunately. And that's also because you need people who are experienced not only in biotechnology, but also in cryptography and computer science. So you need people that are able to blend 3 different industries together. And what's worse is, and this is where it gets really hard, you need specialist skills and specialist laboratories for it. Now, thankfully, there are people that are doing it. So, for instance, the military in Tel Aviv are actually using DNA cryptography for all of their storage, and there are certain big tech companies that I will not name that are also using this and working on this for their internal storage. It's very cool. If you want to know who they are, you can ask me afterwards. And then at present, we also know that the US military is also using it to do a lot of their encryption for very secret confidential files, which is awesome. Or not, depending on how you feel about them. Now, the biggest issue with DNA cryptography and research is that no one seems to be able to agree. So you have an issue where, unfortunately, you have Snea over here and a man over here, and you want to use RSA, and you want to use 3DES, and none of us can kind of come to a solution. There's no streamlined agreement as to how DNA cryptography will work or how we should be going about doing it. And there just isn't enough money that's being put into it, which is why I decided to do this talk. Mainly because if we can get more companies on board who are interested in doing this research, we can actually speed up the timeline and create DNA cryptography that can be used for normal everyday users. Now, we get into my favorite part of the talk, which is also the more sci-fi part of the talk. So put on your Spock ears, be prepared to be teleported. Scotty's about to take you on a journey. So what happens if we, for instance, wanted to use DNA cryptography for personal use? What if I wanted to use my personal DNA to encrypt something on, say, I don't know, one of those little keys, and that would be my key for life? I would never have to use a password again. That would be amazing. Well, it's a bit hard. It's a bit difficult. What we found, firstly, is that, for instance, if you are having cancer treatment or if you are exposed to any kind of disease, your DNA will change. So theoretically, what could happen is, say, for instance, we created a personalized DNA key for you on Tuesday. Great, Tuesday. Then on Friday, Tayla gets HIV. And now her DNA has changed. So in theory, her key would change. So we actually have to figure out a way of creating a personalized DNA key that would use a part of the nucleus that goes unchanged, regardless of disease or exposure. The other issue is if you have cancer, your nucleus will change. So you'd go from having a healthy nucleus to one that is mutated. So essentially, if we have personalized DNA encryption, we would then have to question whether or not we'd be leaving those who have diseases or going through treatments at risk. Would they then have to go back to the old way of doing things where they have to remember 50 passwords? I certainly don't want to have to do that. And the other thing is identical twins. So thankfully I don't have a twin because there's already enough of me in the world. But the thing is that identical twins have the same DNA makeup because they come from the same father, same mother, especially if they came from the same egg, etc. So what, for instance, if we had something like an evil twin attack, but in the version of like people, where there's Tayla and there's Tanya, and Tanya decides that Tayla is like, they're going to wreak havoc on each other, right? So that's also something we're trying to understand is how do we create non-identical keys for twins using the same DNA? How do we do that? Now, the answer is that some nucleuses in DNAs in twins can be different, but that's really labor intensive. And the other thing is that it's still really expensive to do this research and this kind of technology. So there are big IT companies that I won't name that are currently doing this research because what they're trying to do is they want to be at the precipice of DNA cryptography because they kind of want to be the first ones to say, hey, by the way, Snea, you no longer have to ever use passwords ever again. You can now use your DNA. Or can you? So then we get into the bigger problem, which is the ethics. Do we even want to do this? So if I take you back to Cambridge Analytica, back those days, it was great. Everyone had their data stolen. We all questioned our lives. Now, how would we feel about a data breach involving our own DNA, where our own DNA sequence could be stolen? Granted, DNA sequences are a lot harder to read than like your password and your email from your grandmother in the 70s, but my point remains the same. And the other point is that our DNA is everywhere. Look at the cups and the plates and the glasses that I put over here. Your DNA is everywhere. But the question is, would I be able to say, take a piece of my hair? Ow. Would I be able to take a piece of my hair or someone else's hair and use it to decrypt their data? That's a huge question and a part of research that we're still trying to understand and figure out. And hopefully we will in the future. Okay, so there was a lot of reading that involved in this talk, a lot of questioning of life decisions, as I said before. But now I'm going to open the floor up to a few questions. And yeah, thank you for listening to my babble. I appreciate it. Here we go.
Hey there, thank you for the talk. It was awesome. So I have a question now. You've obviously showcased how easy it is to get someone's DNA. Now, to solve this problem, could you potentially use the DNA in combination with some other biological values to build some form of biological passport that wouldn't be as easy to steal?
So that's a really great question. So what we did in the early phases of research is we looked at like DNA from a public library and we took one DNA string. And then what we did is we would then add an algorithm to it on top. Or we would add like a salt to it as well. And that kind of helped with in terms of creating something that looked a bit like a biological passport. Unfortunately, we're still in the very early stages of this research. So I can't give you a yes or no answer to your question, but we're hoping in the next 3 to 5 years that we'll have answers like that for you. But for now, what we've realized is if we use something like an algorithm that we have now and add it to the current encryption that we've done, it could be a solution.
Thank you.
You're welcome.
Hello. Thank you for your talk. It was very interesting.
I'm grateful. I'm thankful.
I have remarks and questions, but I try to. So first, the thing is with having your DNS and password is a bit problematic because every 10 years, because telomeres got cut off, you don't have the same DNA. So I'm not sure this would really work. Also, you could forget any witness protection program because you can't fake your DNA unless you can totally rewrite it. But my actual question is, are there actual symmetric or asymmetric cryptographic algorithms in DNA, or do you just map our cryptography and encode it in DNA then? Because this looked a bit like that at the moment.
Yeah, so like I said before, it's still in the very early phases of research. We're still kind of beginning to understand it. So what I will say is at the moment what we've done is we take, say for instance I have, sorry, what's your name?
Dennis.
Dennis, cool. So say for instance I take Dennis's DNA. I'm just going to use it as an example if that's okay. Okay, cool. So I'm going to take, thanks Dennis. So I'm going to take Dennis's DNA, and then from there what I'm going to do is I'm going to add a public DNA library which could house probably over 500,000 people on top of it. And then that's why the encryption and decryption takes so long. And then from there, we then add what's called an algorithm to it. So we'll use like an AES or a DES to it. At the moment, yes, it is very rudimentary. It's not exactly like super cool and a smart way of doing it, but we're getting there. The point is that like if we don't try, we won't know. And in terms of your earlier remarks, in terms of like witness protection and that sort of thing, this is probably why I've also decided to do this talk. Is because if we're aware of these issues now, and we're aware of these pitfalls and challenges and social questions, then we possibly have an answer. We'll be able to react to those issues as they happen. It's no use saying, oh, well, this is going to be a problem and walking away from it. If we actually talk about it now, we can come up with the solutions in the future, if that makes sense.
Thanks.
Alternatively, what we could do is, because we already have public DNA libraries, an alternative, and this is just me like spitballing here, we could assign someone in a witness protection program with a synthetic DNA key for the purposes of their growing life. But something that has no match to an actual human. Because what we did with DNA storage is we created synthetic DNA strings based off of the current strings in process. I'll talk to you, Dennis, don't worry. That's what coffee's for.
Thank you for this talk.
There you are.
Brilliant topic, and very informative presentation, and very well exposed. Thank you for that.
You're welcome.
I have a few questions, but I will keep it very short. You mentioned that we need to think in strings, and the example that you reported uses 3 pairs in order to encode a string. 3 pairs with 4 characters actually make 64. I wonder, if you would use 4 pairs, you would get 256 variation, which is exactly a byte. Why not go there?
So I don't have that answer for you because I'm still thinking. But what I can tell you, let me just go back one. Please hold. Your call is important to us. We will assign you to your next relevant agent. There we go. Right, you are now 1 in the queue. Congratulations, you have made this pit stop. So as you can see here, so what happened here is the substitution cipher. Right. So we knew, regardless of the depth or breadth of that algorithm, that it just wouldn't be secure. So whether we add more or less, it doesn't make a difference because as it stands, a substitution cipher, it's a bit like, I mean, it's a table. Like,
oh, this is fine. I fully understand that for the substitution part. I'm saying that here, you said in the slides before, we have to think in strings, and I think maybe 2 slides before, I don't remember.
No, sorry, there we go.
So we have to think in strings, which makes sense because it's a sequence, and I fully understand. I'm saying that 4 of those pairs could encode a byte 1-to-1. So in principle, having a data set could be actually mapped in any sequence of 4. So that's why I'm not sure that because also in the data we have byte sequence, right? And in principle, those could be mapped by using 4 pairs.
Yes. So we could definitely use the 4 pairs. So what I quite like about DNA is that it won't be your typical TA, TA, GC, TC. Like, no DNA sequence is the same. So for instance, and that's what kind of makes us unique. And so whether we encode it with 2 pairs, 4 pairs, that doesn't really factor into it. It's based off our different DNA strings as they stand. So for instance, my sequence will look a lot different from your sequence. So when we add extra base pairs to it, we could do that. And that's what we've done with the public DNA library libraries, where we actually added those libraries to our already existing DNA that we had on site.
Okay, so basically, if I understand correctly, you're saying that we cannot add an arbitrary sequence to what we have.
Well, yeah. Maybe after maybe during lunch or something, I'll talk to you about it. Okay. Sorry.
Thanks. I have a question related to the storage aspect. What are the error rates, or in general, what are the durability statistics of DNA-based storage?
So, as I said before, we've had many issues with doing DNA storage in terms of DNA contamination, DNA leakage, and then just overflow. Because obviously, when we did that initial research, we didn't have the computational power. So we were doing it off 3 crack machines when we actually did this specific synopsis. And what we realized is that we didn't have the computational power to actually do it retroactively. So it would take us probably a few years to get to the point where we'd be able to do it, and a lot of time and research and money to do it. But we already saw issues in terms of DNA contamination and leakage in terms of like normal data. I'm happy to talk to you more in detail if you'd like. If I didn't properly answer your question, I do apologize.
Okay.
Am I done now?
I guess then the rest of the question will come to you in your coffee breaks.
Danke schön.
So, thank you very much.